Okay, let's get started. So that paper for discussion is an introduction to some of you of the use of phylogeny and ecology and evolution in some areas. What I want to do in this final lecture is talk about a more statistical issue, but one that many people encounter who wish to do something like compare species. This is the last lecture. I know that you're all receiving emails from UBC saying, evaluate this course, evaluate that course. We have no time for this. But it would be useful if you did take some time to make a few comments on the course, especially what would be useful for me is just your thoughts on how the format worked and which topics you thought might be disposable and which ones potentially could be added. Sometimes I think this very last one I could maybe give this lecture, but then the workshop could be a choice of workshops. Maybe one would be phylogenetic data, another might be spatial data, where that might be used in some labs, and but actually faces or confronts similar statistical issues. Anyway, if you have any thoughts like that, write them down in a review. What worked, what didn't work. All that is useful. It'll help future students. So I'm going to start with an example of species data. What I mean by species data and comparisons between species, and trying to learn something about biology, and ecology, and evolution of a group of animals. Okay, so this is a water strider. The back row just woke up here. The two water striders, in fact, a male on top of a female. And this was a comparative study of 15 species. And the biology of the study was one look at a conflict between the sexes over mating. So, you know what water striders are? You've seen them in sort of still water. They skate around. Males spend a great deal of their time chasing females, who spend a great deal of their time fleeing, chasing males. And then if a male does grasp a female, she initiates a series of leaps and rolls and somersaults that can frequently toss him off. Males of some species have evolved clasping genitalia that allow them, with the result that they stay on longer, but females of these species often have counter spines or devices that make it difficult for those devices to clasp and so on. And then mating takes place if the female stops struggling before the male is tossed. So what I want to do in this example is correlate something that the authors did, the average duration of female struggles across species with the average mating frequency. So these are all measured on individuals brought to the lab, measured under controlled conditions, and the comparison of duration of mating and mating frequency was of interest. And a plot of the data looks like this. So along one axis, arbitrary, is the average struggle duration in seconds, and then the female mating frequency, the number of matings per hour. And we can see that there's a positive correlation between the two. So the longer the mating struggles, the higher the mating frequency. And so we're interested in saying something about the strength of this correlation and the standard error for that correlation. But species data have a problem that we haven't encountered with any other kind of data that we've analyzed so far in this course. And that is that species are not independent. And what I mean by that is that they're not sort of randomly sampled from some underlying common distribution. And the reason for that is that species are related to varying degrees by phylogeny. And basically what that means is that some species are very closely related to each other. And other species are much more distantly related to each other. And we can see that there's a pattern whereby a general tendency for this particular trait, average struggle duration, to be similar between similar species, which is, I think, pretty remarkable for a behavior which you might think would be evolutionarily fairly volatile. Here, there's a tendency for closely related species to have similar values. So there's an example. And the flip side of this is that more distantly related species have more distant values, and as a consequence, phylogeny influences correlation between observations from different species, and so they're not independent. And this tendency for closely related species to have similar values is referred to as phylogenetic signal. So basically, the tree is providing some information on the degree of resemblance between your data points in their values. And the fact that we can do that means they're not independent. And all of the methods that we've learned in our undergraduate careers and all the methods that we've learned so far in this course assume that we have a random sample. And a random sample means, well, firstly, every point has an equal chance of being selected, and the other is that selection is independent, the selection of observations. And in this case, closely related species, the presence of closely related species and distantly related species violates that assumption. So the first question that we might ask is, how bad is it? How frequently do we find this tendency for closely related species to be disproportionately similar in the kinds of traits that we typically measure? And one metric for measuring that is called Pegel's lambda. It's a metric that varies between 0 and 1, and basically measures the correlation between close relatives. And so in this survey of the literature, Freckleton et al. Calculated phylogenetic signal for a whole pile of studies, and this is the result. So Pegel's phylogenetic signal is high if Pegel's lambda is close to 1, and phylogenetic signal is low if Pegel's lambda is close to 0. And the upshot of this graph is that large values are very common. Modal value is between 0.9 and 1. So phylogenetic signal is extremely common. I don't understand why it's bimodal. Why the next most common bin should be between 0 and 0.1. If anyone has an explanation for this, I'm all ears. So why is phylogenetic signal a problem? So here's a graph from Felsenstein, 1985, which was a paper published in the American Naturalist, and as far as I know, remains the single most cited paper ever published in the American Naturalist. I can't remember who told me this. Maybe I mentioned this to you already before, but Felsenstein has 4 papers with that status. So four of the papers that he's published in 4 different journals are each the journal's most cited article in all of history. So he tends to publish method papers, and he tends to do an extremely good job of explaining how they work. So here's the paper in AmNat, a classic. And to illustrate this, Felsenstein presented this scenario. Here's 2 traits, X and Y, and we're interested in the correlation between those 2 traits. And each of these symbols represents a species. So there's data on 40 species, and it looks like there might be a correlation between these 2 traits, X and Y. And if we did a conventional correlation analysis, we would probably find an association that's moderately strong. But Felsenstein said, let's illustrate the potential problem with what he called his worst case phylogeny. And it looks like this. So species 1 to 20 are all related by descent more or less simultaneously, and 21 to 40 also, but these two groups of species fall into two clades. So two groups, each of 20 species. And now back to the scatter plot, we've now identified those two groups by symbols. And we can see that there is a correlation between X and Y. But the overall picture is one in which X and Y appear to be correlated sort of once in the phylogenetic history of this group, and then never again. There's no association between those traits within each group. So this would represent an extreme case in which the non-independence of phylogeny is creating a correlation on the basis of a much smaller sample than you think. So here again are the 15 species of water striders, and what the assumption of independence really means when applied to species data. When we just carry out an ordinary correlation between two variables using conventional statistical methods, we're basically assuming that the phylogeny looks like this. That's the only way in which the evolution of every pair of species is independent of the others, since they're common ancestor. But of course, real phylogenies don't have that property. So Felsenstein's solution was as follows. First, he said, let's assume that the evolution of traits is mimicked by continuous random walk, Brownian motion. So Brownian motion is what's often used to describe the movement of particles, in a room, say. And it doesn't actually mean that trait evolution is random or by genetic drift. It's just a mathematical equation that attempts to mimic the trait evolution over time. And one feature of Brownian motion is that the difference between any two species in a trait has a probability distribution that's normal. It has a mean zero, and it has a variance that's proportional to the time since their common ancestor. And this is just an illustration of what Brownian motion might look like in three species that split here and then split here again to produce those three. What Felsenstein observed is, in this case, if we can assume Brownian motion, then A, B, and C are not independent. However, the difference between A and B is independent of the difference between C and the average of A and B. And so he called those contrasts. And they are since known as phylogenetically independent contrasts. And so from a phylogenetic tree of n species, we have n-1 independent contrasts that we can still use to calculate a correlation between any two of their traits. And so Felsenstein presented the calculation details, and it's not that important. He showed it in this case, I'm not going to go into the details. He showed in this case for a phylogeny in which the branch lengths are not all, do not all meet at the tips, in order to illustrate that under Brownian motion, the expected variance of the difference between any two species is proportional to the sums of the branch lengths that connects them. And that these independent contrasts can be standardized by dividing the contrast by its expected variance. And so Felsenstein showed how you could do this using all the data on the phylogenetic tree. then the method involves converting the data on both traits to their independent contrasts using the phylogeny, and then calculate the correlation between the independent contrasts. And so here's a cutaway of the water strata tree that gives some intuition for how it would work. So here's the value of the two traits that we were interested in: struggle duration, mating rate. And so the claim, Felsenstein's claim, is that this contrast is independent of this contrast, is independent of this contrast. And then to calculate these contrasts, we calculate the difference between these two species, the difference between the average of these two species and this third species, and then finally the difference between this species and a weighted average of those three. And the direction of the contrast is arbitrary. We could take 10.24 minus 16.58, or we could do the reverse, 16.58 minus 10.24, but we just have to calculate the same way for both of the two traits. That's the only crucial thing. So Giletti minus Ingrinsis for both traits, then we're good. And so here is the results. Now, instead of the n measurements on each of these two traits of the 15 species, we have 14 contrasts. So we have the contrast in struggle duration, the contrast in female mating frequency, and we see that there remains a positive correlation between the two after correcting for phylogeny. Also, because the direction of the contrast is arbitrary, as I said, we could take this minus this for both traits when calculating this particular contrast, or we could do the reverse. And what that means is that it's possible to calculate all the contrasts so that, say, that it's positive for x, or calculate all the contrasts so that it's negative. All the values are negative for x. And what that means is that because the direction is arbitrary, this line will pass through 0, 0. And you can do all this using the 8 package in R. So it's been around for a while. Emmanuel Paradis created this package years ago, wrote a book on it, gave it to the world for no cost whatsoever. So that's the method of phylogenetically independent contrasts. And what I want to do also is introduce you to an equivalent method that's actually even better. And the reason it's so great is that it's a linear model. And we know how to do linear models now. One of the reasons why I start this course with linear models is that so many of the other methods besides linear models are so much like linear models, like generalized linear models. And here I'm going to introduce a linear model method called general least squares. And it turns out to be mathematically equivalent to phylogenetically independent contrasts. And GLS was invented long ago, long before Felsenstein, 1985. And it was developed for the situation in which the residuals in a linear model, the residuals in Y, between the observed and the predicted values, are correlated, and they don't have the same variance. And we can fix this by taking into account those correlations. If we could know what those correlations are and what the unequal variances are, then we can incorporate into an otherwise straightforward linear model a sort of a weight matrix of these unequal variances and expected covariances between observations. and essentially correct for it in carrying out a linear model of y on x. And we can't just calculate their covariances from the x and y traits. We need to have some kind of a model that tells us what they are or what they're expected to be. And in this case, we know what they are or what they're expected to be. If we can make the assumption that Felsenstein made, that evolution is mimicked by a random walk, a continuous random walk, Brownian motion. In that case, we know what the expected variances are. They're proportional to time. And we know what the expected covariances are. They're the amount of shared history between any pair of species. And we can calculate that from the tree and then apply GLS. And the beauty of modeling this in the linear model framework is that we get stuff out of it that we already use and find useful, like AIC if we want to do model selection, for example. So I thought I would just give you a little bit of intuition for what this phylogenetic covariance matrix looks like for a simple case of 5 species that are related to varying degrees by phylogeny. So under Brownian motion, the expected covariance between 2 species is the proportion of total history from root to tip that they share. So a telase shares this with those 3 species. And so 0.38 is the fraction of the tree that a telase shares with the other 3 species, and so it's just 0.38 all the way across. And along the diagonal, a telase with itself is 1 because it shares with itself the entire histories. So this can be done between all possible pairs. So Pongo and Homo, that's Chips and us, have a high covariance. And what that means is that the contribution of these 2 data points to the correlation between any pair of traits would be sort of down-weighted because they are so similar to one another. It is expected that they would be so similar to one another. And so each of them would essentially count for less. Right, there's Pongo and Homo. So they share, in this case, most of their phylogenetic history up until the most recent split. And this covariance matrix then becomes a weight matrix that is used in straightforward linear model using this GLS function. But again, it makes the same assumptions as phylogenetically independent contrasts. So the main assumption is that evolution in each trait mimics a continuous random walk in time, Brownian motion. For this assumption to be met, the rate of evolution actually has to be constant through time and along all branches of the phylogeny. And we also have to assume that speciation and extinction are unrelated to trait values. These assumptions are actually difficult to verify. Also, I'll show you in workshop this week how the branch lengths of the phylogenies can be transformed to improve agreement with the Brownian motion assumption should the rates of evolution change over time. But there's been a lot of simulations done with this method, and Harvey and Rambaud asked the question, if the assumptions are not met, then is it still good? Should we still use these methods, or should we just go back to the ordinary correlation between traits ignoring phylogeny? And the answer is, in extreme cases, it's actually worse to use the phylogeny if the violation from Brownian motion is severe. So keep that in mind. So it becomes important to evaluate the assumptions of Brownian motion, but as I said, it's actually very difficult to verify those assumptions. And this method that improves the fit to Brownian motion is one that makes use of lambda, which expands or shrinks the branch lengths between nodes over time. And so, it's possible at least to fiddle a little bit with the assumption of Brownian motion and improve the fit. And we'll do an example in the workshop this week. So the 8 package in R can find the best estimate of lambda for a given data set. So those are the, that's the basic method for continuous traits correlating X and Y. And an advantage of general least squares, or GLS, is that we can model continuous variation in a Y trait and a discrete X variable. We can do phylogenetic 2-sample t-tests. We can do phylogenetic ANOVA using the GLS method. And so that sort of extends it beyond the realm of 2 continuous traits. But what happens if there are if both traits are discrete? Does the same issue arise? And is there a solution? And the answer is yes and yes. So the data set that I'll show you is from lilies. So you all know about lilies, but what you might not know about lilies is that generally the showiest lilies are the ones that tend to occupy the sunniest open environments, and or they flower before the trees above it leaf out if they're in the forest. Whereas species that flower in low light environments of the forest understory under a leaf out, such as the Clintonia, then they tend to have small and inconspicuous flowers, whitish or greenish in color. So there seems to be an association between habitat and flower characteristics. So here's data on 17 lily species from this paper, where the flowers were classified as occurring in open or shaded habitat, and whether they had showy or did not have showy flowers. And it's almost a perfect association. But the reason I'm showing this example in the first place is that the number of evolutionary events underlying this association is not necessarily very large. So the showy ones are here shown in yellow, and the inconspicuous ones, or the less conspicuous ones, in green. And here's the phylogeny of the 17 species. And then the background shading I've used to indicate whether they occur in shaded habitats or open habitats. And what you can see is that most of the showy species are related by descent. And that the number of evolutionary events in prehistory needed to produce an association between showy flowers and open versus shady habitats is not very large. How many times have showy flowers, so it looks like inconspicuous flowers is ancestral, and showy flowers have evolved only twice in the history of lilies. And there have been only 3 transitions between habitat types, twice into open habitats and 1 back. Now, that's not necessarily the exact evolutionary history, but it's sort of a minimum number. And the point is that how many independent evolutionary events are actually represented in this tree, and knowing how few events actually took place, or how few transitions took place between these environments, how strong is the evidence statistically for an association between flower showiness and habitats. So again, it's the same problem that Felsenstein described and proposed a solution for. And Pagel proposed a solution for discrete traits as well. And you can think of it as somewhat analogous to the continuous case. It assumed that evolution in each trait again mimics a random walk in time. But this time it's a discrete random walk in time. So species through time occupy one of, in this case, just two states, and that there's a probability at any point in time that they'll transition from one state to the other. And those rates can be estimated from the data. And that process of a discrete random walk is called Markov process. You can think of it as somewhat analogous to the discrete analog of the continuous random walk, Brownian motion process. So it's all likelihood based, and now you know what that is and you're comfortable with it. And what it does is it models the transitions in both of these traits through time, and then asks whether the transitions are associated. Is there a tendency, when a transition in one trait occurs, does it tend to be associated with a transition in the other trait as well? I'm not going to go into detail, but I can point to a package in guess what language that will carry out these calculations for you. Now that I've shown these problems to you, and I have pointed to the solutions to these problems, I'm also going to tell you about the fly in the ointment. So this is still a fairly active area. So Felsenstein, published a paper in 1985, Pagel's test in the 90s, and people have been using this method ever since. And Wayne Madison and his brother have also been involved in the development of some of these methods, some of the predecessors for discrete traits. And Wayne and Rich Fitzjohn, who was a PhD student of Sally's, published a paper in 2015 in which they pointed out that Pagel's test is actually, it doesn't actually work. And how they showed this was that if you had a phylogeny in which only one change occurred in both of two characters, and they occurred more or less in the same place in the phylogenetic tree, then Pagel's method is liable to give a statistically significant result. And this is a bit worrying because I already pointed out the problem in the lilies that very few transitions need to be incorporated in order to produce this very strong association between these two traits. And now they're showing that actually you can get statistically significant associations between traits on the basis of only a single transition in both of the two traits, if they happen more or less at the same time. Well, you could ask, what are the chances that they would occur at exactly the same time? And that's what, and the answer is chances are very low if the rate of evolution is low. But humans are incredibly good at picking traits that tend to co-occur at exactly the same time. We first look at all the traits and all aspects of the biology, and then we look at those that tend to have a strong association, and then we carry out Pagel's test. And so through this combination of inadvertent bias and Pagel's method, you can get significant results when the association is not real. So they call this within-clade pseudo-replication. Part of the problem with Pagel's test is all of those instances in time in which neither trait is changing, that also represents evidence in favor of there being an association, right? Because neither of them is changing all that time. So Pagel's actually right But we don't think evolution works that way. We count more in terms of transitions rather than instances in time. So that's the nature of the problem. So there are no clear solutions yet, but actually solutions have emerged and are currently being tested that seem less susceptible to this problem. Now I'm going to another UBC person was involved in another paper that actually started to point out issues with, not so much again with the method, but the idea that evolution actually behaves according to a Brownian motion process. Biologists likely have a favorite example of a lineage that has evolved something spectacular, such as devilishly horned lizards that squirt blood from their eye sockets. You knew that, right? Or marine sloths that grazed ancient seabeds. It's hard to know what to do with those types of events when modeling their evolution on phylogenetic trees. But their point was that these singular, interesting events in the history of life really don't jive well with the assumption that evolution mimics a random, continuous Brownian motion. Method development continues apace. Again, a method has been suggested that might solve this problem, so stay tuned. Almost surely it's implemented in R already. What I thought I would do at the end is just, I guess, underline this. I don't know if you've had a chance to hunt for packages to do something you want to do to analyze your data in R, but at CRAN, which is the main repository for contributed packages in R, people maintain these task view pages. So last week, I opened up the task view page for phylogenetics. What if you were interested in finding out whether these new methods, which are based on hidden Markov models, are actually implemented in R? You can go to the task view, and this generous soul has decided to take on the task of curating this page to keep it up to date, which, as you can imagine, is quite a task because there's new contributed packages happening all the time, and revisions of those packages that add new functions. And then some packages just fade away when their authors decide not to continue them. So I want to just show you what a task view looks like for randomly chosen subject phylogenetics, since it was the topic of the last lecture. Getting trees into R. So here's a series of packages that will allow you to actually take text-based phylogenetic trees and import them into R. And so this ape will do it, and that's what we'll use this week in the workshop. But there are other packages for other functions that will do it, or will help you deal with different phylogenetic tree formats. Here's a paragraph on all the packages with utility functions for manipulating phylogenetic trees in R, or associated data associated with the species on the phylogeny. There are packages that attempt to estimate the ancestral states, the traits possessed by ancestors of the modern day species, using models of evolution on phylogenetic trees. There are packages out there that will analyze diversification, estimates of speciation and extinction rates. One of my students even wrote one. You could write one. Estimates of divergent trend, estimating the trees themselves to begin with. Time series, paleontological data can also be incorporated. And here's some packages. Tree simulation packages exist, including Geiger. models, packages that will help you model trait evolution on phylogenetic trees. Just look at this. So there are different methods, new methods are appearing all the time. I'm looking for, you see hidden Markov model on here yet? Look at that Markov model. I'll bet this is already implementing the sort of the new methods that are far more robust than Pagels for discrete traits. Trait simulations, tree manipulation, community microbial ecology uses phylogenetic trees. Phyloclimatic modeling. It just goes on. Phylogeography, population species delimitation, tree plotting and visualization, tree comparison, taxonomy. Gene tree, species tree, interactions with other programs, notes. You get the picture. I've made my point. There's an embarrassment of riches in R. And all of this is contributed by good people who charge absolutely nothing for it. And I hope that this course has given you at least up to the first step in sort of exploiting this universe, because it's incredibly useful. You will, before long, need some method which we didn't cover in this course. And it might only have been invented last year, but chances are there's already an R package for it. If it's not on CRAN, it's on GitHub. It's almost essential if you create a new method that you create a program that will implement it, and more often than not, the program will work in R. You'll get a chance to use some of these phylogenetic methods in the workshop this week. I'll be here. I hope to see you there. The course itself gave you, there's two ways I could have done this course when I created it. One was, go deep into linear models and a small number of other topics. But the other thing that I decided to do instead was give a somewhat superficial introduction to a variety of related methods, starting with linear models and showing you, at least intuitively, if not mathematically, the connections between linear models and so many of these other methods. And then also give you a little bit of guidance as to how the methods work and how to interpret and avoid some misinterpretations of the use of these. You're going to need these methods in your thesis, and when you do, all the R tips will still be there, all the workshops will still be there. You might think, oh man, I got to do generalized linear models. I'll go back and do the one we already did, go through it again, and then try it on your own data. So I'll do my best for as long as I breathe to keep the R tips website going. I mean, presumably R can be replaced someday too, in which case you're on your own. Most of you will stick with R. It's hard to avoid because a lot of the methods that you will want to use aren't available in other packages. Certainly, a lot of the conventional packages will have linear models and stuff like that. I remember a few years ago there was a grad student here who came up after the course. I was on her PhD committee as well, and she came up to me after the course and said, well, that was really instructive, but I think I'll go back to my menu-based statistical analysis package. And I said to her, that's okay, I'm not taking it personally, but at least I want you to know what you should expect from those other packages if you go there. They should be good. They should do as much as R does. So be critical, and if not, come back to the world of R. Look around the table. These people will be in the halls for some time to come. They've suffered through this course just as you have, and already you guys have been helping each other make progress through the workshops, and I hope that continues. I think there's already an R group been established, start one yourself or a data analysis group. It's a way to keep being informed and to bring your problems and get suggested solutions. I'll be around for a while. I'll be happy to see you in the hallways, and I'll wave. And I'm on your committee. I'll certainly ask how your data analysis is going. So thanks for participating, and hope to see you on Thursday. If not, I'll certainly see you in the future. So thanks very much.
I'm sorry. Was this? I thought you meant that.
Yeah, leave me a second.
That will be great when I have to do it.
When the break came.
No pressure.
Hey, dog. I make you.